Very happy today to have Julia Wang joining us from Florida State University College of Medicine, Department of Biomedical Sciences, and the title of her talk is Molecular Mechanisms of Human Pancreatic Islet Dysfunction under Overnutrition Metabolic Stress. A little bit about her. She received her PhD from Johns Hopkins and completed her postdoctoral training with Klaus Kestner at UPenn. She pioneered using high throughput, high content, sorry, just letting a few people, and single cell resolution platforms, including single cell RNA-seq, single cell mass cytometry, and imaging mass cytometry to investigate the molecular states of human pancreatic islet cells. She established her independent research group at Florida State University in 2019, and her lab applies genetics, genomics, and bioinformatics approaches to understand the molecular mechanisms of diabetes. So far, her lab has co-authored over 10 publications, including 6 primary research articles, with Dr. Wang as senior or corresponding author, and her research has been supported by NPOD, Breakthrough T1D, and the ADA. I'm going to drop the paper in the chat right now, and please add questions there, and she'll field them at the end of the talk. Thank you so much for joining us today.
Thank you, Monica, for the kind introduction. And this is my first time presenting here, but I want to say I'm a long-time follower for sugar science, especially the Friday weekly review and then the think tank platform where really we can hear the most updated science about diabetes research. So thank you, Monica and the team to really establish this platform.
Thank you again.
Thank you. So today I'm going to tell you guys about our recent work on the molecular mechanism of human pancreatic islet dysfunction under overnutrition stress. So to just bring everybody into the same page, type 2 diabetes is our diabetes of interest here. And this diabetes is take almost more than a decade to develop, generally believed. The clinical onset of the diagnosis of type 2 diabetes is correlated with the increased blood glucose levels. But before this clinical diagnosis, there's a lot of things going on, including the insulin resistance in the peripheral tissue and altered insulin secretion in the beta cells, and then differentiated beta cell mass. I want to bring your attention to 2 prominent environmental factors here. One is the increased glucose, the other is the increased adiposity. These 2 factors, if not contributing to diabetes, but definitely it's like one of the proximal events directly related to the diabetes onset and beta cell dysfunction. So combined effect of elevated glucose and fatty acid is coined the term as glucolipotoxicity. Glucolipotoxicity has been studied in beta cells for over decades now. It's believed that this combined high glucose and high fatty acid function on beta cell to induce heightened inflammation in the beta cells and also increase their oxidative stress and impair autophagy. However, some key questions remain, including what is the effect of glucolipotoxicity on other islet cells, considering all the cells they are facing the very similar environmental factors similar to beta cells. And number 2 is what's the molecular program underlying glucolipotoxicity in beta cells. So towards this, we applied a single cell RNA-seq approach on human islets isolated from 3 female donors and 3 male donors. We cultured these islets under control, 5.1 millimolar glucose condition or glucolipotoxicity corresponding to 25 millimolar glucose and 0.5 millimolar palmitic acid conditions. And then we performed the single cell RNA-seq after that. This project is spearheaded by talented grad student Snow and postdoc Li Guo in my lab. So below these 2 graphs is showing that this system, our glucolipotoxicity treatment, works. As you can see in here, each dot represents 1 donor with connected line represent samples from the same donor. So you can see the glucolipotoxicity condition appear to increase the 3.3 millimolar the insulin secretion under 3.3 millimolar glucose, and then blunt the insulin secretion under 16.7 millimolar. And these results can also be shown as a stimulation index, which is a ratio of the insulin secreted under 16.7 millimolar divided by 3.3 millimolar glucose. And again, the GL condition, you can see a significant reduction in the insulin secretion. It's really showing that beta cells, they physiologically, functionally respond to our heightened glucose and pancreatic acid treatment. And at the single cell space, the first step is we annotate the cell types we have. And everybody is probably very familiar with this UMAP, where each dot represents one cell, and the cells with similar transcriptomic profiles are clustered together. And we can identify all the major cell types inside the human pancreatic islets, and each cell corresponding express their canonical cell type markers. So the first question we ask is whether glucolipotoxicity treatment uniquely impact one cell type versus the other. And our idea, if this is the case, then we should see the alteration in the cell type proportions in our sample under control, under GL conditions. And here on the right is a bar graph showing in each donor what the relative cell type proportion under these two conditions. And we did not see consistent changes in one particular cell types, meaning that at least at this very broad level, we do not see one unique cell type that seems to be particularly vulnerable or resistant to glucolipotoxicity. Looking at this cell type proportion, one thing I also want to point out that for majority of the donors, alpha cells seem to be the more abundant endocrine cell types, at least in our single-cell RNA-seq workflow. This is really highlighting the unique cellular composition of the human islets and really showing the islet heterogeneity. So next, to study whether molecularly, whether some cell types have a specific more response to the glucolipo conditions, we perform differential expression analysis under the assumption that if one particular cell type is very vulnerable or uniquely responsive to this glucolipo toxicity treatment, we will see more transcripts that's up or down regulated. And here, reassuringly, we see beta cell is the #1 cell type that has the most genes that are upregulated and downregulated under glucolipotoxicity condition. Interestingly, the second cell type that mostly influenced by this overnutrition treatment is ductal cells, really showing that, again, it's not this glucolipotoxicity condition is not uniquely sensed by beta cells only, but all the other pancreatic cell types, including the isocrine cell types, also responsive to these environmental alterations. We also check the molecular pathways that are overrepresented in these differentially expressed genes. And here, focusing on upregulated genes, we can see several pathways, such as response to ER stress, organonitrogen compound metabolic response, or response to oxidative stress, programmed cell death. Several pathways seems to be enriched in those upregulated genes across the board, right? Again, to reinforce the idea that the majority of the endocrine pancreatic cell type, if not all the endocrine cell type, they are responsive to glucolipotoxicity treatment, and they mount very similar molecular profiles, at least from the pathway level. One thing about the differential expression analysis is that this process, including the compare two groups, can potentially influenced by number of cells we have for each group. As you can see here in epsilon cells, we actually didn't detect any genes that differentially express, but this may be because we didn't have that many epsilon cells. To mitigate this potential effect, we use an orthogonal machine learning method, agar, to further prioritize cell type. So this agar is basically treat this cell type prioritization as a classification task. The underlying idea is if this treatment really significantly alter the transcriptomic profiles of the corresponding cell type, then this classifier agar can really separate the cells of their treatment or control purely based on their transcriptomic differences. And if this algorithm works perfectly, you will see the separation AUC is 1, meaning that this machine can 100% always accurately predict whether this corresponding cell type is under control or GR conditions. Or if the algorithm doesn't work well, you will see the AUC is more or less close to 0.5, meaning that it's now in here just like a flip of coin. The machine cannot guess whether the cell is under treatment or under control conditions. And using this machine learning algorithm, you can see again the top 2 cell types that seems to be very good classified in the algorithm is DUCT1 beta cells, right? Meaning that glucolipotoxicity really changed a lot of significant transcriptomic changes in these 2 cell types to make them separable compared with the control status. And another interesting observation is the alpha cells, another endocrine cell types, they don't appear to have a lot of differentially expressed genes, and they also don't seem to have a huge transcriptomic shifts under glucolipotoxicity. So this drives us to ask whether these molecular changes in alpha cells is reflected in the alpha cell functional changes. Towards this, we measure the glucagon secretion in alpha cells under control and GL conditions, and we observe no differences, right? In both control and GL condition under both 1 millimolar glucose or 3.3 millimolar glucose condition, glucagon secretion seems to be indistinguishable in alpha cells. Hence, we can reach our conclusion, alpha cells are resilient to glucolipotoxicity, at least under our experimental context. And this alpha cell resilience is also a recent concept. I think there's multiple groups also working on that, showing that even in the early stage of type 2 diabetes, alpha cell transcriptomic shift seems to be the minimum across multiple other endocrine cell types. Next, we study the cell-cell interactions inside our pancreatic islets, right? Again, under the idea that glucolipotoxicity seems to impact a lot of pancreatic cell types, we wonder whether this glucolipotoxicity also impact the cell-cell interactions. We here focusing on the paracrine hormone signaling because that's one of the most prominent hormone cell-cell interactions inside the pancreatic islet. And we use the algorithm called cell-CHAD, which is inferred paracrine hormone signaling based on their ligand receptor expression levels. So the top graph is showing the outgoing signaling pattern, meaning that where the source of different paracrine hormones. And you can see this graph is, there's 2 major findings from this panel. Number 1, our cell type annotation is pretty good, right? Because the glucagon is send out the source of glucagon as alpha cells and source of insulin is beta cells. This is really aligned with our understanding of the biology. And secondly, compare with control, the GL condition significantly reduced the insulin signaling and interesting ghrelin signaling, right? So again, the insulin ghrelin signaling source is significantly reduced under glucolipo conditions. And we can also look into the recipient end using the incoming signaling pattern inference. So this incoming signaling pattern inference is based on the corresponding receptor expression in different cell types. For example, for the pancreatic polypeptide, the receptor NPY1R is uniquely expressed in the pancreatic epsilon cells. We can hence derive this link that the recipient for pancreatic polypeptide is epsilon cells. In the incoming signaling pattern part, we also can observe several findings. Number 1, most of the endocrine targets seems to be restricted, contained within the endocrine compartment, except insulin. Insulin appear to target more promiscuously in all the pancreatic cell types. As a matter of fact, the highest recipient for insulin appear to be the pancreatic endothelial cells. This also aligns with our biological knowledge about this insulin signaling because the insulin receptor is one of the receptors is more universally expressed in the different pancreatic cell types. Number 2 is we compare with control, the GL condition significantly reduced number of incoming signaling, especially in the insulin paracrine pathway and the ghrelin pathway. So based on this outgoing and incoming signaling pattern, we can reconstruct this paracrine interaction map. Again, based on our single cell data, we can infer the ligand and receptor pair. So we know where is the source. For example, insulin is sourced from beta cell, and where the target of insulin. And here we show that insulin target mostly alpha, epsilon, and pp cells. We can also further see that under glucolipotoxicity conditions, several paracrine signaling patterns reduced, mostly is on the insulin end and on the ghrelin end. So this is really showing again the glucolipotoxicity not only changed the transcriptomics in individual cell type and elicited corresponding functional change, but they also changed the cell-cell interaction. So there's a systematic response under these overnutrition conditions. Using single-cell RNA-seq, we can also perform a pseudo-time trajectory analysis. Here I borrow one slide from my graduate students. The idea is if you take a picture of the rose field where there's a rose in different stage of blossoming, and you can align them based on their similarities of reconstruct the entire process of how a rose goes through a flower bud to advent to blooming. Similarly, in the single cell context, because we have the single cell cellular state of each individual cells, we can align the individual cells based on their transcriptomic similarities, and then hence we can help to reconstruct the cellular trajectory under our external treatment. In here, our external treatment is metabolic stress. So using the single cell trajectory analysis, we focus on beta cells. We found that beta cell trajectory seems to separate the beta cell cluster into two leaves. One leaf of these beta cells are enriched in the cells derived from control conditions as shown in green here, and the other leaves of the beta cells enriched for cells in the GR condition. And these two leaves is also corresponding to pseudotime with earlier pseudotime values, a cell with earlier pseudotime values to later pseudotime value. So these results showing that our trajectory does reflect our external treatment of GR, right? So again, control cells more enriched for the cells with earlier pseudotime values versus GR conditions are enriched for cells with bigger pseudotime values. So using this pseudotime label, we can now study the dynamic gene expression along pseudotime. So here we look at all the genes that are dynamically regulated along pseudotime, and we further cluster these genes, and we see there's three major patterns of dynamic gene expression emerge. Cluster 1 are enriched for those genes that upregulated during the earlier or middle stage of the pseudotime, and pathway analysis showing that these genes are enriched in pathway that corresponding to responsive to ER stress. And cluster 2 genes are showing the mostly immediate early or middle stage expression along the pseudotime, and these cluster 2 genes are enriched in pathway corresponding to phosphorylation, post-translational modification, or chromatin remodeling. And cluster 3 genes display a bimodal pattern. The pathway enriching cluster 3 genes are more corresponding to the metabolic pathways. So here, using this dynamic gene expression pattern, we can reconstruct the molecular events after glucolipo condition, glucolipo treatment. Basically, immediately after the external stress, cells mount the ER stress. This is followed by epigenetic or post-translational modification and other cellular signaling events, and culminate in the metabolic and other cellular state change. And as I mentioned, cluster 2 genes are enriched for biological process related to the epigenetic regulation and post-translational modification. We took a closer look at the molecular functions of those enriched molecular functional pathways in these cluster 2 genes. We found one particular molecular function term, histone H3 methyltransferases are enriched in the cluster 2 genes. So histone 3 methylation, here's a graph summarizing the different modification. There's a lot of loci that's methylated by different methyltransferases and demethylated by different demethylases on histone H3. We searched the literature and found 7 different small molecules that targeting different histone methylation loci and targeting different methyltransferases or demethylates. And our goal is using these small molecule inhibitors and perform targeted drug screen. Towards this, we use the human endosheath beta H1 cells because this is a cell line, this is a human beta cell line that allows us to have more controlled condition compared with the primary human islets, which is very variable, had a lot of biological heterogeneity. However, for the endosheath beta H1 cells, it's well known that it's not very sensitive to glucolipotoxicity because they have very heightened sterol CoA and other metabolic process related to the metabolizing of the fatty acid. So we first perform a titration experiment to see at what concentration of the palmitic acid, this lipid, we can really elicit a corresponding functional reduction in these endosheath beta H1 cells. And we found that a combination of 25 millimolar glucose and 2 millimolar palmitic acid is enough to reduce the stimulation index in these human beta cells. Just as a reference, in the primary human beta cells, 0.5 millimolar palmitic acid is already sufficient to show a glucolipotoxicity effect. In endosheath beta H1 cells, we need to increase the concentration fourfold. Using these systems, we performed this targeted drug screening, and we found that among the 7 small molecule inhibitors, only A366 seems to have a further exacerbation effect when in combination of the glucolipotoxicity. And this is shown by under the combined A366 treatment and the glucolipotoxicity treatment, beta cells have further increased baseline secretion, baseline insulin secretion under 3.3 millimolar glucose, and further reduction in the 16.7 millimolar insulin secretion under 16.7 millimolar glucose. So A366 is a molecule that targets A3K9 methyltransferase G9A and GLP. We first want to check whether these 2 enzymes indeed regulated somehow under glucolipotoxicity conditions. So we go back to our pseudotime and then extracted the corresponding transcripts corresponding to G9A and GLP, and we see that as expected, these 2 enzymes are upregulated in the middle stage of the pseudotime. And these 2 enzyme expression patterns also appear to be highly correlated with each other. Next, we find the output of these 2 enzyme activity, which is A3K9 dimethyl, also is significantly upregulated under the GR condition compared with control. And on the left is the images of the H3K9 dimethyl staining, and on the right is a quantification. Furthermore, we show that A366 treatment, which is the inhibitor of the G9A and GLP, significantly reduced the A3K9 dimethyl level. So the combination of A366 and the GL treatment can move the cells A3K9 dimethyl level almost the same as their control conditions. To understand what's the transcriptomic changes correlated with this H3K9 methylation dosage, we performed bulk RNA-seq on the human beta cells treated with glucolipotoxicity condition or control condition, and with or without the drug A366. And here on the left is a PCA analysis where each dot represents 1 sample. And we can see that PC1, which explains 59% of the variance, separate the samples of control versus GL condition, versus PC2 separate the samples based on their drug treatment, A366 with or without the A366 drug treatment. Next, to compare whether the gene expression profile changes under the GL treatment show similarity or differences of the gene expression changes under the drug treatment, we perform this analysis called RRHO, rank-rank hypergeometric overlap. And here on the graph on the right help to guide the interpretation of this RRHO. If these two different perturbations, in here our axis is the perturbation of glucolipotoxicity, and on the y-axis is the perturbation of the drug. If these two perturbations have concordant effect, we should see a lot of signals on the concordant axis, which is in the B and C quadrant. However, if these two perturbations showing change the gene expression in different directions, we should expect to see their signal in their discordant quadrant, which is in the A and D quadrant. And this is actually exactly what we see in our experiment, right? Just to reiterate, in here from this RHO plot, we can see those transcripts that's upregulated in GL appear to be downregulated by the A3688 treatment, right? So to summarize what we know so far, glucolipotoxicity increase A3K9 methylation. This we already see from the staining levels. And inhibition of the A3K9 exacerbate the glucolipotoxicity effect. This we show by treating the beta cell with A368, which is inhibitor of A3K9 methylation. Their beta cell insulin secretion function is worse. And the inhibition of A3K9 methylation reverse the transcriptomic changes in beta cells under glucolipotoxicity. Right, to sum all this up, here comes our conclusion. A3K9 methylation appears to play an adaptive role in beta cell under glucolipotoxicity. And if you block this adaptive increase in methylation of A3K9, beta cell fare worse under glucolipotoxicity. And in the last few slides, I want to say another new area we're working on. From this plot, we can actually see the A3K9 dimethyl appear to show bimodality in the endothelin beta H1 cells under control conditions. And this bimodality is further, the entire bimodality shifts up under GL conditions, right? So this seems to indicate beta cell naturally have different epigenetic states as dictated by A3K9 dimethyl levels. These findings before that, so we want to check whether this A3K9 dimethyl bimodality is correlated with cell cycle. Towards this, we use the image stream where all the cells go through the flow system, and then there's a camera there to take picture of each cell as they go through the flow. Using this multi-parameter imaging stream platform, we can specific subset the endothelin beta H1 cells on those cells in the G0, G1 stage. And then here, again, we confirmed even when we subset cells in a particular cell cycle phase, we still observe bimodality. Some cells naturally have higher A3K9 dimethyl level versus some cells have lower dimethyl level. And these differences in A3K9 dimethyl is not correlated to their insulin expression or their DNA staining. This finding really echoed one of the earlier findings from the Andrew Pospisil's group. They show that epigenetic dosage seems to be correlated with the functional distinct beta cell subtypes. In here, they are using another repressive marker on histone A3K27 trimethyl to separate beta cells into the higher functional group and lower functional group. And we're seeing the A3K9 dimethyl may be another separate either orthogonal or independent epigenetic marker that help to further distinguish beta cell subtype. I also want to bring out this another recent paper published by Sanjita's group. They show that DNA methylation is also seems to be stochastic and appear to separate beta cells based on their different function group. So in summary, using an islet glucolipotoxicity model, we examined the overnutrition effect on individual islet cell types. We answered two questions we showed early on in the first slides. Number one, what is the effect of glucolipotoxicity on other islet cell types? We showed that islet cell types, they have varied sensitivity to glucolipotoxicity. Beta cells and ductal cells are the most sensitive to glucolipotoxicity, while alpha cells appear to be resilient. And we also further delve into the molecular program underlying glucolipotoxicity in beta cells. We show that A3K9 methylation appear to play a protective role and is increased in beta cells under glucolipotoxicity. We also show that epigenetic dosage is important to rewire the transcriptomic landscape that help to beta cell respond to their metabolic stress. And overall, our conclusion is epigenetic mechanism being most heritable and reversible offer promising avenues for biomarker discovery and precision therapy in type 2 diabetes. That's all for research. And last but not least, I want to acknowledge everybody involved in this work and everybody in my lab. This work is spearheaded by Snow and Lee in my lab, and Lee has subsequently moved on to her new position, and other graduate student, research assistant in my lab. This work also is in collaboration with multiple collaborators in FSU and with Wang Lee in Cincinnati Children's Hospital. And I want to thank my founding agents, and also the islets and tissues from this research is acquired from IIDP and NPOD. And I also want to thank all the islet donors and their families, because without their selfless contributions, our work cannot be performed. Thank you for your attention. I would like to welcome any questions.
Thank you so much. Beautiful work. Just a quick comment from Sarah Richardson in laboratory. She had to run, but she wanted to say that this work is really beautiful work, and congratulations, Julia. I wanted to open it up to anyone else who would like to either unmute themselves or drop something in the chat to ask a question. I guess I'd like to sort of direct the talk a little bit towards type 1, of course, because that's our focus. But your data kind of highlight, or they highlight glucolipotoxic stress-induced transcriptional and epigenetic remodeling in beta cells. Do you think similar stress signatures, particularly like involving the ER stress and the UPR genes, are evident in residual beta cells from individuals at early or pre-symptomatic T1D stages? And how might you go about capturing those?
Yeah, definitely. Thank you for the question. So the ER stress is generally believed to be the common pathway between type 1 and type 2 diabetes. And this idea has been, I think, early on by Desiree Ezrick from Belgium and several other groups, showing that remaining beta cells in the type 1 diabetes also suffer from very high level of stress. Regarding how do we study this, actually there's a recent paper from Michael Stiezel's lab in, I think it's from Jackson Laboratory. What they do is they have a parallel treatment, either some islets treated with the ER stress inducers, epigargen, and then some islets with the different type 1 interferon cocktails, and then just study their transcriptomic changes and epigenetic changes, and then they can compare the similarities and differences between these two processes. So I think this approach is something we can take as well. One thing I want to say that is some of our findings, such as like alpha cells resistant to the stress, definitely apply in the type 1 diabetes. I hope they apply to the type 1 diabetes context, because we know alpha cells is one of the robust cell types that doesn't seem to be undergone the same immune attack in the type 1 diabetes as in the beta cells, and they're also surviving the different metabolic stress in the type 2 diabetes conditions.
But how might you use your protocol approach to kind of further inquiry in that space? I guess that's what I'm asking. I mean, if someone saw your work today and was like, oh, that's really cool, let's do a collaboration,
Yeah, I think mostly the further study, what I'm thinking is on the molecular comparison and then find the
Right.
common or dissimilar pathways because our
Yeah,
workflow.
the
um,
methylation pathways, I think, are the most interesting, right? So you could kind of see, and I really liked your the pseudotime trajectory analysis. So like if you could apply that to work done in other labs or being done in other labs where they're looking at these alterations in upregulated and downregulated genes in context of type 1, right? So that would be really cool.
Yeah, one definitely, and one part I didn't show is we actually can directly project our findings into the GWAS studies, trying to directly use our finding to explain whether we can sort of prioritize some of the GWAS genetic signals using our system. Um,
Yeah.
we can also project into the both type 1 and type 2 to see whether we can find very key molecules or pathways that's involved in this process.
Right. I liked also, when you were kind of walking us through the altered ductal cell genetics and the plasticity. Right, you saw the altered transcriptional and epigenetic changes in the ductal cells under this overnutrition stress. So, like, given emerging evidence of this ductal to endocrine transdifferentiation, could similar ductal remodeling happen in T1D, maybe like as an attempted regeneration pathway, or like, a source of when that's happening, neoantigens that trigger immune sort of recognition. Like that whole ductal pathway is very interesting. And
Definitely. Yeah.
and if you're talking about like overnutrition too, like how similar does the mirrored hypoglycemia or glycemia that happens when beta cells are not functioning properly mimic that overnutrition?
Yeah, yeah, to really study the temporal orders of different cells and how the crosstalk, that I think that the holy grail, that's what we want to do. And
Yes.
using inductive cells is also, there's another area we are very interested in, but we haven't spent much time on it, is we do know there is a connection between diabetes and pancreatic cancer. So
Yeah.
diabetes is like beta cell dysfunction, and pancreatic cancer, the cellular organ,
Type 2.
right.
Yeah.
Yeah, so this area we can also see whether there's some common shared molecular changes in different under different environmental conditions, and see whether we can explain the pathology.
Yeah. Yeah. We just had Catherine Maidler from Germany on. She had, she sort of discussed her YAP and pancreatitis work. And that kind of implicates exocrine ductal inflammation in initiating type 1. How do your ductal cell findings intersect with that concept? Like, could metabolic or viral stress in ductal cells contribute to this whole pancreas inflammatory phenotype seen in type 1? What do you think about that? I mean, I know I'm pressing you to the limit here because I know you're focused on type 2, but
I think definitely, but one thing we were trying to look for is, because it's known that this glucolipotoxicity in beta cell, they do elicit this immune response, but we actually did not see much immune signatures in beta cells or in other cell types. But this system is very complex. I think depends on the treatment duration, dosage, and everything. You really can potentially see different things. I think really our system is very flexible, and the bioinformatic approach is really even more flexible. So what we are trying to do is to collect a lot of data from different systems, from different platform, where we can really integrate everything together and perform this molecular trajectory analysis, so that we can really see how that the cells progress from normal to disease condition to different disease, and maybe they can crosstalk. So that's our really, in the future, that's what we want to do.
I have here's something from Jemming Liu, City Hope in LA. Great talk, really nice work. Have you had a chance to integrate your data set with the HPAP T2D data for trajectory analysis?
Yeah, very good question, and congratulations with your paper, Goming. I think I just saw you did something similar with the trajectory analysis. We actually try to do something in-house as well, but one of the major challenges is different data come from different sources have huge batch effect. And then when you're trying to just force them together, how do you mitigate this battery effect will still preserve the biological signature is one thing we still need to figure out. Yeah, but definitely HPAP is one of the data sources we want to leverage on, and also NPOD. I see there's a lot of data published recently on single cell RNA-seq on their data collection as well. So that's what we want to do.
Yeah. I guess I'll circle back one more question with type 2. Have you thought about looking at samples from or cells, human cells, sort of post GLP-1, administration? So for instance, here's a very heavy person who then went on GLP-1 and then, became a donor. You had access to these. That would be a very interesting signature to look at, right, for you.
The very interesting signatures, very challenging to guide, because I think to really make all the analysis very sound, ideally you want before and after. So that is like almost infeasible from human donors.
So then you have to go into the dish in vitro.
Badly going to the dish. And regarding that, I think there's also a recent paper from Zhuo Xianmeng's group in the Zhejiang University. They actually exactly did that. They treat their mouse with the GLP-1 semaglutide, and then they study the transcriptomic and the epigenetic changes.
Yeah, and there's also been sort of limited, but still some use of GLP-1 in type 2s. I mean, sorry, type 1s. So that's another place to look for signatures, and maybe the mouse model is the first place to go. But yeah, your system's really beautiful, and congratulations. Well trained in the world of the Kestner lab in Penn. You can tell.
Thank you.
Yeah, excellent. Really nice data. And so thanks again for sharing. And we look forward to, the next body work coming out of your lab, and perhaps some cross-pollination with the T1 world.
Thank you so much, Monica.
Thank you, Julia. Thanks again. Have a great rest of your day. Bye-bye.